I am Devendra, lecturer in botany, Government Degree College, Kagajnagar. Today we discuss about structure of microsporangium and also so called as TS of Anther. This is a very important question for foremost question in degree level. So generally this type of in the angiosperms, the flower is the reproductive organ. In the flower having the four circuits, out of the four circuits, the outermost two circuits are like as a calyx and corolla is a non-essential organs, and the androecium and gynoecium layers circuits are essential organs. So this type of androecium and gynoecium is the directly participated in the reproduction due to this one, the very essential organs and also called as reproductive organs. In the androecium is the male sex organ and also gynoecium is the female sex organ. First we see in the androecium having the various type of plants having the various number of anthers. This type of anthers having the various types, some of the dithecous and also some of the monothecous also. Generally this type of anther looks like as a bilobed and dithecous also. But in between of the dithecous having the one longitudinal one septa is formed in between of the two lobes. Due to this reason, the cross section, the anther looks like as a tetragonal structure consisting of four microsporangias, each lobe having the two microsporangia. Due to this reason, looks like as a four microsporangia, looks like as a tetragonal structure. When the mature, the macrosporangium, later the two macrosporangia of each lobe fuse and form as a one pollen sac-like structure. Due to this reason, this type of is called bilobed and dithecous also. This is the structure of structure of anther in the young stage, having the looks like as a four. Actually, this type of the two lobes, but the one septa is formed in between of the two lobes. Looks like as a four lobe structure. Due to this reason, looks like as a tetragonal structure, having the four macrosporangia. But when mature the anther, the two lobes fuse and form a one pollen sac-like structure. So this type having the bilobed and dithecous also. But some of the species like Malvaceae having the monothecous, in between of the monothecous and the single lobe, in between of the lobe having the one pseudo septa is formed. Due to this reason, looks like as a bilobed. But when mature, the pseudo septa is ruptures and form as a one single lobe structure like structure in Malvaceae members. Due to this reason, this one looks like as a monolobed and monothecous in Malvaceae members. Remaining all are most of the species are complete dithecous and bilobed. Next, structure of anther or structure of microsporangium. This type of microsporangium is a circular and surrounded by the four layers. Generally, this type of microsporangium having microsporangium, when we cut the cross section, tangential section, looks like as a four lobe structure. Each lobe covered with the four layers. One is the layers are epidermis, endothecium, middle layers, and another one is the tapetum. These are complete belongs to the parietal layers. The outermost layer protect the outermost layer, the one called as epidermis. This type of epidermis is single layered. This type of single layered epidermis the outermost, this type of layer protect the pollen chambers and help in the descent of anther to replace pollen. Generally, this type of the outermost epidermis having the very thick walled cells, but some part, especially some of the cells is specific part having the very thin walled cells. Due to this one, that part is called as stomium. When mature the anther, the pollens are produced in the microsporangium. When the pollen grains are matured, the place of the stomium, the anther is descends. Due to this reason, the pollens are released out. So this type of outermost layer participate in the releasing of pollen grains or microspores. This one is the when this type of the matured anther. When the matured anther at the place of the stomium part, this is the epidermis. Due to this reason, the inner side of the pollen grains all are released out through the stomium place. So when form next one, the inner side of the epidermis, uh, this general this type of epidermal layers, the outermost layer anther, which is single cell in thickness. The cells of the thin layer divide only anticlinally in order to cope up with the growing anther. In the inner side of the epidermis, another one layer is present. The type of layer is called as endothecium. So this type of endothecium, the next to epidermis, in the inner side of the epidermis, the endothecium is present. The cells of the endothecium are radially elongated and develop cellulosic fibrous bands in their inner and tangential walls. Due to this reason, this type of enlarge endothecium complete having the large size of cells, and these cells come having the cellulosic and fibrous bands. When the microsporangium mature, slowly the endothecium cells all are ruptures and these all are complete disappears at the time of the mature microsporangium. In the inner side of the endothecium, another 1 to 3 layers of middle layers are present. This type of middle layers in the inner side of the endothecium, this type of in middle layers, all of the disease generate at the time of the meiosis in the microspore mother cell. So at the time of the before the meiotic division, this type of the middle layers all slowly disappears and form as a one chamber-like structure in the inner side of the chamber. The microspore mother cells divide meiotically and produce the large number of applied microspores. In the inner side of the middle layers, the last lower side layer is called as tapetum. It is the innermost layer of the anther wall. So it is composed of single layer of cell characterized by the dense protoplasm and prominent nuclei. This type of cells remaining all parietal cells, the tapetum cells are very large in size and having the prominent nuclei also, and dense cytoplasm is present in the tapetum cells. Depending upon behavior, tapetum is of two types. One is the amoeboid tapetum and another one is the secretory tapetum. So this type of amoeboid tapetum, see this type. Next, this is the A is the amoeboid tapetum and another one is A is the secretory tapetum and B1 is the means the D is the amoeboid tapetum. So first we see the amoeboid tapetum. It is A very primitive type of tapetum in the lower most of the cells in the angiosperms having this type of amoeboid tapetum. This type of tapetum, the complete during the drying up process of anther, the periplasmodium hydrates and deposits as a tryphan on the wall of the pollen grain. So this type of amoeboid tapetum complete supply the food materials to the spore mother cells and microspores. And another one is the secondary tapetum. This type of secondary tapetal cells remain attached to middle layer till the development of pollen grains. It is more common among angiosperm. This type of tapetum is present in the advanced of angiosperm plants. This type of tapetum participate in the various type of functionings. One is the this type of tapetum, it provides nourishment to the developing of pollen grain. Supply the food materials to the growing pollen grains or microspores and before the meiotic and after the meiotic divisions. For the developing of microspore mother cells and also after the completion of the meiotic division and supply the food materials to the microspores or pollen grains. It helps in the formation of enzyme. This type of pollen grain produces the various type of materials like as sporopollen. This type of material form as a exant type of layer on the pollen grain. This type of tapetum helps in the transport of food materials to inside of the anther. So remaining last one, the tapetum helps in the formation of the pollen ball also. This type of some materials of the tapetum participate in the formation of the pollen ball. This is the tapetum functions in the various type of plants. Next one, the sporogenous tissue. Sporogenous tissue in the inner side of the parietal layers remaining the very thick walled and also very large cells is called as sporogenous tissue. This type of primary sporogenous tissue give rise to the microspore mother cell. Before the formation of microspore mother cell, the sporogenous tissue divide and produce the microspore tissue. This type of microspore tissue divide and meiotically produce the microspore mother cells. This type of some of the sporogenous cell remain non-functional and serve the food materials for the developing of microspores. So, large quantity of sporogenous cells are slowly divide and produce the microspore mother cells, and remaining some of the sporogenous cells not participate in the division and form as a microspore mother cell. This type of all are non-functional sporogenous cells. This type of sporogenous cells act as a supplying food materials for the growing microspores. So this type of in the angiosperms, the microspore mother cell is the diploid stage. This type of diploid microspore mother cell undergoes meiosis to form a microspore tetrad. Generally, this type of microspore tetrad, when formed after the formation of the microspore tetrad, which separate out of the microspores or pollen grains. The process of formation of microspore from microspore mother cell is called as microsporogenesis. This type of process is called as microsporogenesis. When the formation of the pollen grains or microspores, the process is called as microsporogenesis. The process of formation of microspores from the pollen mother cell through the meiosis is called as microsporogenesis. So generally this type of the cells of the sporogenous tissue undergo meiosis to form a microspore tetrad arranged in a cluster of 4 cells. As each cell of sporogenous tissue has a potential to form a tetrad, so each cell is microspore mother cell. On maturation and the dehydration of anther, the spores disassociated and develop into pollen grains. The pollen grains released with the dehiscence of the anther. So generally this type of spore tetrads form as a various type like as a tetrahedral and isobilateral and decussate and other some of the T-shaped and linear structure also. So this type of each microspore mother cell divide meiotically and produce the spore tetrads. This type of spore tetrads, the spores arranged as a various type like as a tetrahedral. Means the spores, four spores, microspores attach with the one and another and form as a tetrahedral manner. In sometimes this type of the spores all are arranged four spores arranged as a isobilateral also. And another some of the decussate also means the lower side of the two spores arranged as a one side and remaining two the upper side of the two pollen grains arranged as the opposite manner of the lower side. This type of is called as decussate manner. And another some of the microspores arranged as a T-shaped structure. T-shaped structure means the lower side two cells are collaborate, one undergoes another, and the upper side of the two cells arranged as a perennial due to longitudinal due to this reason looks like as a T-shaped structure. This type of transport tetrad is called as T-shaped. And last one is the linear shape. The four sporogenous spore cells arranged as a one linear manner, arranged as a one single layer like structure and the one above one. Due to this reason, this type of is called as linear. So the various type of species, the spore tetrads arranged as a various types like as a tetrahedral, isobilateral, decussate and T-shaped and another one is the linear shape, linear structure. This is the microsporogenesis. This is the when formed the microspores in the inner side of the microspore enzyme is called as microsporogenesis. Thank you.